Assalamualaikum and greetings to Madam Wan Natasha Wan Ahmed. First, let me introduce our group members, which is Nur Hirniwati binti Tahir and Nurul Atika binti Suhaili. We are from M38022 or 2E. Group 7 will present about the propagation of chickpeas and rubber tree by using stem cutting. A stem cutting is a separate portions of the stem through vegetative propagation. The stem cutting method ensures that new plant retain the characteristic of the mother plant without any variations. There are a few steps that must be taken when making the stem cutting. Disease and pests, for example, should not be present in stem section, and maintaining high humidity around the cutting is critical. First, I will explain about the propagations of chickpea by using stem cutting. Chickpeas or Cicer Aritinum L is a self-pollinator diploid in the Leguminosae family, and the third largest grain legume cultivated have fluctuated in recent years, with frequent crop failures due to biotic and abiotic stress. The stem cutting have many potential applications for improving the chickpea. Agriculture and horticulture experts utilize techniques such as stem cuttings to keep control of plants' reproductive ability. The objective of this study is to aim and develop a standardized, simple, rapid and reliable method with a high success rate for the propagations of chickpea, C. reticulatum, C. echinuspermum and their interspecific hybrids. Next is the methodology. The material used are rivasane, ethanol, plant regrowth regulator PGR, which is each of the IBA and NAA contains 0.5 mg per gram. Other material used are honey, potting mix, and 4 chickpea cultivars with 5 or more branches. After preparing the materials, we will go through to the procedure. First, prepare 10 cm long cuttings from chickpea cultivars at 2 different development stages, pre-flowering and post-flowering. And then, by using 100% ethanol, sterilize cuttings by immersing 4 cm of their ends. 3 cm of the cutting space was dipped in PGR mix, honey, and honey plus PGR mixture. Dip 1 cm of the bottom end of the cutting into honey, then 3 cm of the cutting base into PGR powder. Use 2 different sterilized cutting substrate, which is rivasane and a mixture of rivasane and potting mix. Then place the cutting into the 10 cm pot. Place the pot on a bench covered with clear polythene and mist every 10 or 15 minutes in 5 seconds for 2 weeks. Each of the cutting was measured by its rooting rate, number of roots, length of roots after 2 weeks. After the procedure is done, we will go through to the result in discussion. The figure shows the effect of several rooting treatments and rooting substrate combination on chickpeas and hybrid rooting rate, root length and root number. The information shows simple column chart with label. Number 1, honey with ribosane. Number 2, honey with sand plus potting mix. Number 3, PGR with ribosane. Number 4, PGR with sand plus potting mix. Number 5, honey plus PGR with ribosane. And number 6, honey plus PGR with sand plus potting mix. Number 4, which is PGR powder and the sand plus potting mix, have the highest rooting rate, root number, survival rate, root length and chosen as the best treatment combinations for chickpea cutting propagation. Sand increases the amount of air space in a potting mix and it includes no infections, fungi or other disease. A sterile potting mix is safer for plants. Number 1, which is the honey with river sand, has the lowest yield for overall yield because making a simple mix for striking cuttings, we need to combine equal parts of compost, coir peat, sand and perlite. The figure shows the reaction in rooting rates and survival rates of various genotypes. A mix of sand and potting mix after PGR powder treatment and growth at the pre-flowering stage. The survival level were between 83.3% and 100% in PGR treated sand plus potting mix substrate after 3 weeks and the maturity of chickpea accessions during pre-flowering phases. Wild accessions 95.8% to 100% and interspecific hybrids high survival rate have also been achieved, ranging from 75% to 100%. ICC 3996 had the lowest rooting rate 87.5% even with the best combination of treatment. In general, pre-flowering more successful than the post-flowering stage for propagation. However, the rooting rate of root number, root length and survival rate were substantial differences in genotypes. In discussion, the treatment of IBA result in the greatest frequency of root formations and the greatest quantity of roots. The addition of NAA to a nutritional solution containing IBA did not enhance rooting rate or root number, but the roots that grew were stronger and healthier than IBA alone. PGR treatments, cutting substrates, and donor-plant growth stage were all key aspects in Sisal species vegetative propagation. The suitable technique for achieving the highest rooting rate and maximum survival was to treat cut stem with PGR powder during the pre-flowering stage and grow them in sand plus potting mix substrate. The genotype also played a significant effect in the response of different chickpea breeding lines, cultivars, and accessions of C. reticulatum and C. echinuspermum. In conclusion, the technique used in planting the stem cutting method to produce efficient, highest rooting rate and maximum survival of vegetative reproduction of chickpeas is treating the stem cutting with PGR powder, 0.5 mg per gram IBA and 0.5 mg per gram NAA at the pre-flowering stage and growing in sand plus potting mix substrate.
Secondly, I will explain the propagation of rubber by stem cutting. The rubber tree or Hevea brasiliensis is a perennial crop and can be propagated by various methods, including in vitro techniques, nursery seedling, budding, and grafting. All of these have their own advantages and disadvantages in plant production. However, stem cutting is a well-known technique that will address the issue of a scarcity of rubber planting materials during the off-season and will also allow large-scale production of rubber propagules at an affordable price for rubber outgrowers. The objective of this study is first of all to ascertain the survival rate of brown and green rubber stem cuttings as affected by stem types and naphthalene acetic acid or NAA levels. Second, to determine the shoot and rooting growth of rubber stem cuttings affected by stem types and rooting compounds. Lastly, to determine survival rate, shoot length and root length between rubber stem cuttings as affected by stem types and NAA at varying concentrations. The methodology. First and foremost, the material used such as two types of rubber stem cuttings, which are brown stem cutting and green stem cutting, with a height about 40 cm and consists of 4 or 5 nodes, naphthalene acetic acid or NAA, sandy loam soil, and manure. The procedure. Firstly, brown and green rubber stem cuttings were prepared from harvested stems for each of the two clones. Second, trim to a uniform length of 40 cm using a budding knife with each cutting containing 4 or 5 nodes. Third, immerse the cutting's basal portions in 0.0, 7.5, 15.0 or 22.5 gram per liter of NAA for 6 hours before planting in nursery bags. Fill with sandy loam soil mixed with manure in a 3:1 ratio. Lastly, the number of sprouted cuttings was counted 4 times at 20-day intervals following planting. The results and discussion. Figure A shows the brown stem cutting of rubber showing shoots 20 days after planting. Figure B shows the brown stem cutting of rubber showing shoots 40 days after planting. Figure C shows the brown stem cutting of rubber showing well-developed shoots 60 days after planting. And Figure D shows the brown stem cutting of rubber showing well-developed shoots 80 days after planting. Irrespective of the stem types and NAA concentrations, none of the green stem cuttings survive even under the same propagation conditions as the brown stem cuttings. On the other hand, rubber brown stem cuttings survive irrespective of the stem types and NAA level. Between brown and green rubber stem cuttings, a significant difference in survival rate, sprouting, and rooting potential was observed. This could be related to both biotic and abiotic factors, particularly excessively high temperatures and very low relative humidity during the early stages of planting, as well as fungal infections on cutting. The reason that brown stem cuttings sprout and grow successfully may be due to the hydrolysis and availability of starch contained in the stem tissues of hardwood cuttings. Furthermore, the reason that none of the green stem cuttings survive could be related to their susceptibility to soil-borne pathogens. To sum up, the rubber brown stem cuttings had a higher significant survival rate than the green cuttings from which none sprouted. NAA's impact on sprouting and rooting growth on rubber brown stem cutting, 50 gram per liter of NAA outperformed better and higher than other concentration. In conclusion, stem cutting can be used successfully to propagate chickpea and rubber, but several factors must be considered in order for stem cutting to develop and grow. In the case of chickpea, the most effective treatments are PGR powder and honey plus PGR mixture. As discussed in the chickpea result, PGR combined with sand plus potting mix is the most effective soil mixture. On the other hand, for vegetative propagation of rubber using the stem cutting method, brown stem cutting is preferable to green stem cutting. This is because rubber brown stem cuttings had a better survival rate than green stem cuttings, of which none sprouted. Consideration must be given to factors such as stem cutting types, growth medium, season, and time of propagation of rubber stem cutting, as all of these factors affect survivability and growth.